Hey everybody, and welcome to Chapter 13. So, Chapter 13, just like the previous chapter, which felt unconventional, this one will also feel unconventional. Also, the textbook is very in-depth on this chapter, way more in-depth than I want you to get into. So think of this lecture as kind of like an outline for some of the things in this chapter that I want you to really focus on. So it goes into a lot of detail about things like sensitivity and just noticeable differences and thresholds and things like that. And I want you to know kind of the big broad picture of that, but I don't want you to commit to like learning all these really nitty gritty type of things. All right, so what are we talking about this chapter? This chapter is about touch. So we're going to be talking about the anatomy. We're going to be talking about the brain. So in the first video, we're going to combine these things and talk a little bit about sensation, which is going to be kind of what's going on the surface of our skin. And then we're going to talk about perception, which is getting to the brain and how the brain processes it. From there, we're going to talk about the somatosensory cortex, so the specific part of the brain that helps us process touch sensations, and then we'll kind of end by talking about pain and sensitivity. Alright, so what is this chapter about? This chapter is about three main ideas. Kinesthesia, which is your perception of where your body is in space. Proprioception, which is kind of understanding your internal state, so kind of knowing whether or not you are moving, partly, but whether or not you are tired, whether or not you are aching, whether or not you are hungry. And finally, somatosensation. And somatosensation is going to mostly be kind of like movement and understanding the combination of all the sensory signals from your skin, to your muscles, to your tendons, to your bones, things like that. Okay, so these three things. Now, you may look at this first one, kinesthesia, and you may ask, wait a second, that sounds a lot like vestibulation. And you're very, very close, or you're right, it does sound like vestibulation, but there's a very fine difference between those two things, between last chapter and this chapter. Last chapter was all about how we maintain balance and how we understand orientation for a specific part of our body, and that specific part of our body is our head, right? Because our inner ear, which is going to take care of a lot of that vestibulation, that's all in our inner ear, right? It's all in the vestibular organ. And so if your hand is upside down, that's not, so you know that your hand is upside down, right? But that's not something that the inner ear is taking care of. That's something that kinesthesia is taking care of. So knowing, without looking how many fingers you're holding up, that's kinesthesia. Knowing whether or not your arm is outstretched, that's kinesthesia. Proprioception, I think of these as things like hunger. So hunger or being tired, that kind of internal state, and recognizing that's proprioception. And finally, somatosensation is most of what we're going to be talking about this chapter. It's like just being able to pick up things and, being able to move around, grab things, understand when things are touching us, things like that. All right, so let's talk a little bit about the anatomy of touch. So we're going to be starting out by talking about touch receptors. And touch receptors are going to be basically a mechanoreceptor. So we talked about mechanoreceptors already this semester. We talked about it when we were talking about the hair cells, right? So these hair cells that when they are pushed in one direction or the other will send out action potentials or will stop firing action potentials. We have very similar mechanisms here with our sense of touch. This time, they're not hair cells. Instead, they are actual cells that when pushed or when vibrated upon, they will open or close their ion channels, which will then facilitate, or it'll inhibit or it'll accelerate the number of action potentials that are firing. So they are mechanoreceptors, which basically just means that they respond to mechanical stimulation. So things like being pushed, being bent, being stretched, these types of things, any kind of the word that the textbook uses and that I was taught was deformation. So anything that deforms the cell. But the word deform kind of has a loaded connotation because it goes back. It's not like it's permanently changed. If it's pushed or deformed in any way temporarily, it can fire out action potentials, which we register as some kind of change on the surface of our skin. And remember, our body, our nervous system, is just a glorified change detector. So we're just, again, looking for changes across the surface of our skin. So the touch receptors that we're going to be talking about, there are 4 main ones, and they are going to be broken down into these 3 different categories. 1, what kind of stimulation is this receptor responding to? 2, how big is the receptive field in which this receptor is responsible for? And 3, how quickly does it adapt? So what is the rate of adaptation? Does it notice very quick changes or very slow changes? And so here's kind of the cross section where you can kind of see these 4 things. So we got our Meissner corpuscle, our Merkel cells, our Ruffini endings, and our Pacinian corpuscles. And those are in the order in which they are deep. So the Meissner corpuscles are right here on the very surface. The Pacinian corpuscles are down in the very deep parts of it. And so you can kind of even just see, just on kind of how these things are shaped, that they are going to have different receptive fields. What does that mean? Well, if you look at the Pacinian corpuscle, it's wide, right? It takes up a lot of area, which means that it's going to have a large receptive field. Does that mean that it's more sensitive? No, it's actually the exact opposite. Because it's only one cell that covers that much, it makes it much less sensitive. Think about if you had 1 cell versus 10 cells that occupied the same area. Those 10 cells are going to make you more sensitive or more better able to detect fine, tiny changes on the surface of our skin. So let's look at each of these in a little bit of detail. We have our Meissner corpuscles, which are right here on the top. These have a small receptive field, as you can see, compared to the Ruffini endings, which are a little bit wider, and the Pacinian corpuscles, which are a lot wider. And they are very fast adapting. So they get used to skin being touched or stimulated very quickly. The Merkel cells, these are also a very small receptive field, as you can see here, these tiny little things, and they are slow adapting. So they take quite some time to get used to skin changes. The Pacinian corpuscles down here at the bottom, as you can see, they have a very large receptive field. They are very fast adapting. They get used to that very quickly. And the Ruffini endings, also a very large receptive field, are very slow adapting. So what is it that these things do? The Meissner corpuscle is responsible for vibration detection, for holding things steadily, and so they're sensitive to skin deformation. What does that mean? Basically, if you were to hold something in your hand, for example, you know that you're holding it in your hand because, in this case, this Rubik's Cube, the flat surface is pressing up against the palm of my hand. And what it's doing, the way that I know that is because it's just pushing up against those cells and deforming them. It's basically squeezing those cells, and when they squeeze those cells, they're firing more quickly. So those are the Meissner corpuscles. The Merkel cells, these things are going to be used for texture perception and being able to understand patterns. And so the most common example here is Braille. So like knowing whether or not if you're able to read Braille, or if you have your computer in front of you, just kind of feeling the keys in front of you and where they are, where the home row is. That is the Merkel cells. The Pacinian corpuscles, these are going to be used for very deep pressures, and the Ruffini endings mostly used for finger position and things like that. They're not only found in the fingers, but that kind of like knowing the stretching of the skin, that is the Ruffini endings. There are other kinds of receptors though, and we're going to go into them in a little bit more detail later in this chapter, but as you may have guessed, by this very restricted definition, we don't only detect vibrations, right? We don't only detect skin deformation, or do we? Can you think of anything else that would qualify for that? There are other things like heat and pain and pleasure that don't fall within this kind of category of vibration and skin stretching. So we'll talk about some of those. But just to give you kind of a quick broad overview, the thermoreceptors, Thermoreceptors are going to be used for hot and cold, and so we actually have two separate fibers of these that we actually have in the surface of our skin and in our bodies. We have two different kinds of thermoreceptors, what's called warmth fibers and cold fibers. And as you might expect, the warmth fibers are looking out for warm change, the cold fibers are looking out for cold change. And so these cells pretty much only respond whenever they detect a change of something in comparison to the surrounding tissue. So if you were to plunge your arms into cold water, that would register as an immediate change, right? But if you hold them down there for a little while and it changes maybe 10 degrees, but it's still freezing, you're probably not going to detect that change, right? And so again, it's all about change detection and and how this compares to other parts of your skin's temperature. The nociceptors, sometimes called the nociceptors, are all about pain detection. So being able to register, detect changes in our tissue that could be considered painful. And so you can think of this as kind of like the body's way of telling your brain that there could be some tissue damage. And so here where it says it transmits information about noxious stimulation, basically that just means anything that could be a damaged tissue. So like, for example, if you get pinched really hard, that registers as pain. Why? Because theoretically that pinch could leave tissue damage, right? It could bruise up, you could tear off part of your skin, you could yank out hair or whatever. So that could potentially be harmful. And because it could be harmful, it really gets our attention, right? And so there are two different kinds of pain. We'll talk about that in just a moment. But these two different kinds of pain, we're going to have some of these receptors that are myelinated and some that are not myelinated. So if you remember back to chapter 1 or chapter 2 when we were very briefly talking about the structure of a neuron, the axon, that long tunnel, that wire that transmits the signal of the neuron, that thing is coated in fat, right? It helps insulate that signal so that it gets from one place to the next. Some of these cells in our pain receptors don't have that insulation. And so I want you to think, why might that be? Why is it the case that some kinds of pain don't require a very fast transmission? So think about that, and we're going to come to that in a video in just a moment. But for now, I want to quickly review some of these different cells, the somatosensory cells. So we got the Meissner corpuscles. Sometimes the book refers to these as tactile corpuscles. If you're getting poked, that kind of change in your skin, that's the Meissner corpuscle. Merkel cells, as I mentioned, is kind of like reading Braille. So those kinds of texture differences for people that don't have sight, that are able to read using tactile sensations. The Pacinian corpuscles, sometimes these are called the lamellar corpuscles. I feel like, to be honest, honestly, if you are reading this, outside of this class, it's kind of a toss-up in which of these you're going to use. It seems like for a long time people used the old lamellar corpuscle, and then people started using Pacinian, and then now it feels like it's kind of going back to lamellar. So if you ever see that phrase, they mean the same thing. These are used for grasping and releasing objects. So like if you're holding on to something, that requires deep tissue pressure, right? And so that is the Pacinian corpuscle. And finally, the Ruffini endings, sometimes called the bulbous corpuscles. These are all about kind of stretches. So think of like finger movements or if you are stretching your shoulders back, that is the Ruffini endings. All right, so let's very quickly talk about the pathway in which we get to the brain. So just like in previous chapters, we're going to have this very quick roadmap. So how do we get from here, our fingertips or our toe tips, all the way up here to our brain? Basically, they are going to go straight from the surface of our skin, the epidermis. They're going to go straight from there to different nerve connections all the way up to the spine. Once they get to the spine, then they're going to go straight up to the brain. So these touch sensations can travel as far as 2 meters, which is a little over 6 feet, right? Because if you think about people that are over 6 foot, they register kind of, sensations in their toe all the way up to their brain, and it feels simultaneous. So it's very, very fast traveling. So it's going to travel up through what's called the dorsal horn of the spine. If you took anatomy and physiology in the biology department, you've probably heard this phrase before as it relates to the spinal column. And so you can kind of see here the dorsal horn is this kind of internal band of fibers that winds its way up through the spine. And in the spine, this is where we start first being able to kind of consider, calculate, and weigh which kinds of sensations are more important than others. So, for example, if you are holding two things, and let's just say, just for the sake of example, that one of these things is hot and one of these things is not hot. Some of that information is going to get to your brain faster than others, right? That calculation is going to take place here in the dorsal horn. So that's before we even get to the brain, right? Which is kind of cool that we're already starting to do some of this perceptual loading here in the spine. And so in the spine, there are actually two different pathways that we can get from the spinal cord to the brain. And it depends on what kinds of sensations we are experiencing. So the two different pathways that we have. The first that we have is the spinothalamic pathway. And this is the pathway in which most of the kind of what I would consider life or death types of information go through. And as you might expect, it's a little bit faster. So the spinothalamic pathway is going to be for temperature and for pain. So these two different kinds of sensations, they are both really important, right? So if you stick your hand in boiling water, you need to know immediately. If you get cut seriously, you need to know immediately that you are in some kind of pain. So that is going to move through the spinothalamic pathway, which you can kind of see in this figure. It's in your textbook. I know my big dumb head is kind of blocking some of that. But basically, it goes straight from the dorsal horn up to a specific part of the thalamus and then straight to different parts of the cerebral cortex of the surface of your brain. The other pathway, though, is the dorsal column medial limbical pathway. That's a mouthful, so usually I'm just going to call it the DCML pathway. Most people do. And this pathway is a little bit more complicated because it is actually going to include lots of information from any other area of our bodies. So anything that's not pain or temperature is going to be the DCML pathway. And so you can kind of see it actually takes a little bit slower to get there because we have moving kind of up in the dorsal horn, up to the cuneate nucleus, up to the medial limbicus, then to the thalamus, and then over to these different parts of the brain. So you can almost think of like pain and temperature as having kind of like an easy pass, where they're able to get on the express roads and bypass some of that traffic whenever there are congested parts. So in other words, like if you are experiencing a lot of touch sensations, you're going to prioritize the painful ones, right? Because it's going to take a little bit of a faster pathway. And when I say faster, I'm talking milliseconds, right? I'm talking very, very, very short but effective, noticeable differences. And the last thing that I want to talk about for this particular video is what's called somatotopical representation, and that is a mouthful, but basically it's an idea that we talked about even when we were talking about vision. We were talking about how our visual field is kind of laid out in the occipital lobe in a very orderly manner. The same is true for our bodies. And so our bodies are represented on the surface of our brain in a very orderly way that has a specific function to it. And so, basically, if you were to touch, for the sake of argument, your elbow, it's going to stimulate a part of your brain. And then if you were to move your finger up to an adjacent, to a neighboring part of your arm, a neighboring part of your cortex is going to be handling that information. If you move up a little bit farther, it's going to move just that much in the brain as well. And so basically what that means is kind of like you can take the human body and kind of lay it out over the surface of the brain, and that gives us what's called the homunculus. And the homunculus is a Greek word that I think means something like little person or something like that. Basically, it's kind of a way to represent the human body across the surface of the brain, such that if I wanted to, I could take an electrode and I could stimulate one part of your brain, and it would you would feel it on the surface of your skin, and then if I move that node just a little bit in either direction, you would feel it on the surface of your skin in either direction as well. It doesn't happen randomly in a different, complete, separate part of the body. It's very orderly that way. And you can kind of see that here reflected, where here, let's focus on the right hand picture for a second, where you can kind of see how the face is laid out across the brain, where we have the forehead up here, then the eye, then the nose, then the face, then the lips. If we were to take an electrode and stimulate, for example, right here, then we would feel that in our face, kind of around our nose, and if we moved up, we would feel it in our nose, and then we would move up, we'd feel it in our eyebrow, then move up, we'd feel it in our forehead. You get the idea. One of the curious things about this that's really great is that you can kind of see, especially if you look in the textbook, it's much more effective because, actually, I think I can move. Yeah, I think I don't know if that's going to be permanent or not, but I just moved the box. And so you can kind of see how the face takes up a large part of the brain, right? Compared to, let's say, the knee and the thigh and the calves, does not take up that much part of the brain. Why? Because we use much more, or we pay attention to much more of the surface area of our face than we do of our leg. Why? Because it's more important. We need to know more if there's something on our face, something that could hurt our face compared to something that could hurt our knee or our calves. If something is in our face, it could be fatal, right? That could be a life or death situation, whereas if it's something that is affecting our legs, we stand a better chance of survival. So, in other words, our brain kind of prioritizes these things based on sensitivity, based on how much processing we do here. We're very sensitive in our lips, right? And the reason why is because we have a large part of our brain, in comparison, dedicated to our lips. All right, I've gone over a little bit of time, longer than I thought that I would, so I apologize for that. I'll catch you in the next video where we talk a little bit more about the somatosensory cortex and how that changes what's going on in perceiving our bodies.